I'm Wendy Herzog, science and peptide enthusiast. In this episode of Exploration Science, Professor Ingrid Dijkgraaf and Dr. Amine Jmel discuss their exploration of tick salivary proteins as potential therapeutics, imaging agents, and targets for vaccines. So first off, thank you both for agreeing to do Exploration Science.
Happy to do so, Wendy. Yeah.
Really appreciate it.
Thank you for the invitation.
Great. So I want to talk about ticks.
Yes. Yeah. Yeah.
So tell me a little bit about, we'll get into that specifically, but maybe Ingrid, if you could start out, just tell me a little bit about your laboratory and what your general research interests are.
Yes. Well, I'm working at Maastricht University, the lab of biochemistry, and we are working on ticks indeed, and mainly the compounds, the peptides and proteins that are produced by ticks. And our whole research department is interested in cardiovascular diseases, so atherosclerosis, thrombosis, and other bleeding disorders. And ticks seem to have very good proteins that could help us with treating these kind of diseases. They inhibit platelet aggregation. They can inhibit blood coagulation. So from a pharmaceutical point of view, these proteins are very interesting.
Yeah, definitely.
Yeah.
And we have a guest with us, Mohammed. I understand you're a postdoc in Ingrid's lab.
Actually, we collaborate with Ingrid. I'm actually working in the biology center of the Czech Academy of Science in Czech Republic. I work, it's in the south of Czech Republic. A city is called České Budějovice. To be precise, it's the Institute of Parasitology. And I'm a postdoc there in Dr. Kocifaki's group. So, and we work mainly on ticks.
Okay. Great.
And we also, as Ingrid mentioned, we also explore these very interesting proteins and peptides from the tick saliva. And that's why we collaborate with Maastricht University to dig into this gold mine, let's say gold mine of peptides. And that's how we met with Ingrid, and we are still collaborating, and we are collaborating for the future too.
Oh, that's fantastic. So what's the so the lab is sounds like it's a parasitology lab and you're focused on ticks. What's the primary goal of the research out of your lab?
Okay, so we are not actually only one lab working on ticks. We are at least 3 or 4 labs working. So each lab is focusing on a different aspect of ticks. Some of them focus on diseases transmitted by ticks, such as Lyme disease. But our lab is focusing on these interesting proteins that exist in tick saliva. And what we do is we explore what's the benefits of these proteins, and we try to apply them to treat some diseases. And the collaboration with Maastricht University and the Department of Biochemistry and Ingrid is to focus on cardiovascular diseases and applications related to platelet aggregation, blood coagulation, and things like that. And we're really impressed by what we can find and discover in this saliva of these small parasites.
So Ingrid, how did you first come across these proteins from tick saliva?
That's actually more than a decade ago. Then a colleague of mine, Pieter van de Vijver, I acknowledge him too less in my papers, I have to say. He came up with a paper because we were searching for compounds that target chemokines, because chemokines play a role in atherosclerosis. Atherosclerosis is considered as a continuous, as a chronic inflammation, I have to say. And we thought, well, if we can target chemokines, maybe we can then stop the migration of monocytes into the plaque. And he came up with a paper where they found chemokine targeting compounds in ticks. And there they described 3 proteins, but there was no structure. There was only some SPR curves, and that was actually it. And then we thought, well, they did it with expression, and it seemed when we read through the materials and methods section that expression was not so easy. We thought, well, let's go for synthesis, because that's what my lab is doing, chemical protein synthesis. And the first compound I started with was pretty easy to synthesize. And that's how it all started. So just in this, we were searching for a compound, and my colleague found it, and there I started.
Right. So it's interesting. I mean, I think maybe just to take a step back and just talk about tick saliva and why it has all these proteins in it. So, ticks are, I mean, maybe you guys should explain this to me, but these ticks have to inject not only immunomodulating peptides and proteins so that they don't get attacked by the immune system, but they have to prevent coagulation and platelet aggregation and keep the system flowing, because they have to feed for, I was reading, I think it's like 2 weeks before they get their meal. Does that sound about right?
Yes. Yeah. Yes, that's true. When I started in these tick fields, in the Netherlands, we have only a couple of tick species. And when I read some papers, I saw that there are over 900 tick species. So you have hard ticks and soft ticks. And hard ticks is the majority, and they can really feed up for about 2 weeks. The soft ticks, they do this within seconds, a couple of minutes, and then it's over.
Right. Yeah.
Yeah.
It's amazing.
It is.
We have lots of ticks here. So every time I go for a walk with the dogs in the summer, when we get back to the house, I do my tick check, because even though they're treated, they still bring the ticks into the house. And then eventually the ticks will die if they, drink their blood. But if they decide to jump off, then sometimes we'll find them on us.
Yeah.
But so I think that something I thought that was really interesting. One of the references that you sent was a really nice review about the structure of different tick proteins.
Mm-hmm.
And so it was interesting to see sort of the diversity of structures, but there's also a lot of similar structures. And so I guess I'm curious to know many things, obviously, but for the first one that you synthesized, the first protein, which of the structures was that?
That was the evasion 3 compound. Okay. That was the inhibitory cysteine knot.
Right. Okay.
Yeah, and that one we could solve with NMR spectroscopy. That was kind of easy, relatively easy compared to the other one. So in the beginning, we were really pampered, I have to say.
Yeah, that's always the way, right? Like, oh, it's so easy to do. But no, not so.
More complex compounds were coming afterwards. Yeah.
Right. Yeah. And the other thing I thought was interesting was that because you had mentioned, the utility of these proteins to act as potential therapeutics, but I also saw that you had looked at them as basically imaging agents.
Yeah.
So maybe you can talk a little bit about that, like the different areas where these can be useful.
Yes. Well, I think here you are really pointing out the options we have with these proteins. That's indeed therapeutics, but also diagnostics. And maybe Amine can also tell something about vaccination, because there we are also collaborating in this field. But I did indeed use this compound for the imaging of atherosclerosis, or I have to say inflamed endothelium. This compound, we synthesized it chemically, and then we coupled one fluorophore to it. And then we did some ex vivo experiments. So what we did is we used a mouse carotid artery. Mm-hmm. And one we activated with LPS, so to induce an inflammation, and the other one we didn't treat with LPS. And then we flew over our fluorescently labeled compound, and then we saw that the fluorescently labeled compound really targeted to the inflamed endothelium, whereas the control didn't. So yeah, we could use it as an imaging agent as well. That was very nice. Yes.
Yeah, that's really, it's fantastic work. Maybe can you tell me a little bit more about the collaboration between your two laboratories?
Yeah, so yeah, maybe I can start and then Amine can add.
Yep.
Yeah, we started a while ago. It was actually at the beginning of the Corona pandemic, and then I came across Amine's supervisor, Michaelis Kotsifakis, and there I saw that they had all kind of ticks, and they were also doing vaccination studies, and Amine and Michaelis are doing also proteomics, and it's something we are not doing in our lab at all. And then we had a Zoom meeting, and there it all started. And I made some compounds because in Amine's lab, in the lab of Michaelis, they have all kind of animal models of tick diseases, and yeah, they have also a tick culture facility, and this is what we don't have in our chemical lab. So I made some compounds and sent them to Amine, and Amine tested them in rabbits. I visited Amine's lab last year, and now Amine is a visiting postdoc in my lab. So he's learning the synthesis, learning NMR and the blood coagulation, and I learn a lot about tick physiology and biology, and also proteomics and all these kind of things.
Right.
So yeah, it's a very complementary collaboration.
Yeah.
So, yeah, maybe Amine, you have something to add?
Yeah. That's exactly the point of this collaboration. Actually, it started with the objective of developing some anti-tick vaccines, but then we discovered that we have more common areas to work on, and we carry on with anti-tick vaccines, but we also try to use peptide synthesis that the lab of Ingrid is really expert on, and we try to synthesize some more peptides for anti-tick vaccines and for other applications too. Right.
Can you tell me a little bit about the tick vaccines? Like, what are you basing those on?
Yeah. So to make it easy, we try to take some of the proteins that exist on the saliva. And we try to inject them into a host, and then the host is developing a reaction against these proteins. So they have antibodies against these proteins, and then once the tick is attaching again, the host is kind of saying, "Oh, I know this protein," and then they have a stronger reaction against the tick, and then we in some cases we see the rejection of the tick. So of course it's not very efficient so far. A lot of research is ongoing on this field. That's why we collaborate with Ingrid. We try to synthesize peptides that are really immunogenic, so they induce a bigger reaction and then a faster rejection of the tick. Of course, the more the tick stays on the host, the more there is a risk of transmitting diseases and so on. So what we try to do is make this as short as possible. This rejection, we make the rejection faster, actually. Yeah.
Sure. But so I know, again, when I was reading some of these publications, I saw there are a lot of proteins that are homologous across species, including with humans. So are these are, I assume, proteins very specific to ticks, and is it still salivary proteins, or is it some other like structural protein of the tick?
Well, actually, there is thousands of proteins in the tick saliva, and these ticks, they are also smart. What they do is a phenomenon called cellome switching. So sometimes they change the proteins that they inject into the host. Okay. So it's not easy to make an anti-tick, but it's not that easy. So we're trying first to understand how the ticks behave, depending on the host, which kind of host. It's not the same when it's a rabbit, and it's not the same when it's a human host. So we try to understand the tick, how it behaves, and then we try to develop some good vaccine that makes, let's say, a universal vaccine, which is, let's say, the dream. It's not really feasible yet, but we dream about this.
Right. Well, that's so. Sorry to keep going down this sort of rabbit hole of tick, vaccines. But I'm also curious. You said that so the tick can actually control which proteins it's putting into the host based on the host. Is there an understanding of the mechanism of how the tick does that, like the biology behind it?
Yeah, actually, that's what we are studying right now. We have some ongoing studies about we call them the transcriptomic and proteomic studies on the ticks. And what we try to figure out is which proteins and peptides they are highly expressed in almost all conditions. So we select these peptides and we collaborate with Ingrid to synthesize them. Once we have them, we try to, again, the vaccination experiments to inject them into rabbits and try to see how these rabbits react to ticks once they have these peptides.
Very interesting. So something, Ingrid, I don't know if you followed the work from Danny Chow and Helena Safavi, but they study cone snail venom, insulins. And so there are these cone snails that will release insulins into water to actually capture fish because they get disoriented from the change in blood sugar. And so I was asking them a similar question about, well, can the cone snail change what it injects into the water depending on, what it's hunting, or maybe if it, wants to get in a fight, whatever it is. And so that's still an area of debate in the cone snail world if they, so it's interesting to see, an arthropod that actually does control specifically. So it'd be really great to know the mechanism and then go out to the rest of the world of venomous animals and see if those mechanisms are similar.
Yeah, that would be very interesting if we can unravel the mechanism, how the tick does this indeed. Yeah, that's very interesting to unravel this.
Yes, absolutely. So in addition to all of these proteins that you're studying from the tick, what about small peptides? Below 50 amino acids.
Yeah, we are working on that as well. From all points of view, so concerning the diagnostics, but also the therapeutics, and also for the vaccines. What we see when we elucidate the structure is that the N-terminus and the C-terminus are generally floppy, or for evasion 3 and evasion 4 and evasion 1, there we see that mainly the N-terminus determines the chemokine specificity of the protein. So it's not really necessary to have the whole protein, and there we can save some time, energy, and a lot of money when we can synthesize a shorter version and when we can synthesize a cyclic version. So we are really focusing on that, so making short versions. And also in the vaccine, in our vaccination strategy, there are all kinds of programs available on the internet that predicts B cell and T cell epitopes. And then you also don't need the whole protein. And what we then try to do is put multiple epitopes on a molecular scaffold to really make a multimer. And by doing that, we can also use different epitopes of different proteins, and maybe also some proteins of the pathogen that's inside the tick. So we are working on that. Yeah.
Wow. Okay. So so I know you study it seems like you study the evasions quite a bit, but what are your focus proteins?
Yeah, for me that's still the evasions. We are still working on that. But I've also been working a lot on Disaggregin. That's an alpha-2b beta-3 integrin inhibitor. So that's involved in platelet aggregation. And there we also want to unravel some platelet mechanisms. I'm not a specialist in this, but some of my colleagues, they are really platelet specialists. So I'm collaborating with them to see what's happening, right? Yeah, with these compounds, with this aggregate. Yeah, so those are the main compounds I'm currently focusing on. So the chemokine targeting compounds and the platelet targeting compounds, and also the vaccine strategy. So yeah.
Can you talk a little bit about the structure of the evasions?
Yes. Well, there are a lot of evasions, but we do see that they are that they can be divided into two families. So the ones that target CXC chemokines, so they are usually having a inhibitory cysteine knot with 3 disulfide bonds, and evasion 3 is one of them. And you also have the CC binding evasions, and those are, for example, evasion 4 and evasion 1. But yeah, there are thousands of evasions, and the question is right now, from a therapeutical point of view, is it good to have a very specific chemokine targeting compound, or is it better to have a somewhat more broadly applicable compound that targets multiple chemokines at the same time? Because yeah, usually when you have an inflammation or an infection, many chemokines are expressed. So yeah, that's really a difficult field. This chemokine world. It's still not known which chemokines are expressed at the same time. So yeah, this has to be unraveled. And also sometimes it's also a little bit how can I say, contradicting the results of this, because some chemokines are really the bad ones and some ones are the good ones. So, and if you inject something in a human being, do you want to target all the bad ones and at the same time also targeting a few good ones? Yeah, you don't know what's good, and yeah, that's a difficult aspect of the research at the moment.
Yeah.
What's the best strategy to follow?
Right. Yeah. Do any of these proteins go sort of upstream of because you could target each individual chemokine, to stop that signaling, but are there any that are sort of looking at the immune pathway that would be upstream of the chemokines?
Yeah, you mean that they inhibit also the cell migration or these kind of things? Yeah, because the platelet of the chemokines we are studying in Maastricht are mainly expressed by platelets, so CCL5 and CXCL4, and they are also expressed by endothelial cells, because we are always in this interface of endothelial cells, inflamed endothelium, platelets that adhere to this inflamed endothelium. Yeah, and what we did see is that when we use evasion 3 or shorter versions, because we were just talking about smaller peptides, so if you use a very short version of it, then we see that neutrophils don't migrate anymore to a chemokine gradient, and also the monocytes, they don't migrate to this gradient anymore. So this is what we see, but yeah, I hope to unravel this further together with a collaborative partner in Leuven in Belgium. Okay. So they are really immunologists, which I'm not. So they can help there. Yeah.
No, immunology is hard.
Yeah, it's very hard, and I'm trained as a chemist. So when I started with all the cells, the neutrophils, monocytes,
it's mind-boggling. But chemistry is much more straightforward.
Yeah, that's true. Yeah.
So Amine, I'm curious. Of all the proteins that you're studying, are there any that you find particularly exciting in the activities that they have?
Yeah, we have actually very interesting candidates and that we are working on. yeah, and they are from different types of proteins. So we have serine protease inhibitors with different ones. So some of them are punids, some of them are serpins. We have also cystatins, so cysteine protease inhibitors, and they are also quite efficient against the immune reaction from the host. Yeah, so those are the two types that we focus on right now, and we have really interesting, and every time we discover something, we are more amazed about what these proteins from tick saliva can do. So yeah, what we are still doing is discovering the mechanistic insights, how these proteins are doing, are stopping the immune reaction, are stopping inflammation, are blocking the migration of monocytes or neutrophils. So it's really interesting. So we discovered that they do that, but we want to know how they do it. So that's we are really focusing on right now.
Ingrid, how about you? Are there any proteins where you were just like, wow, really? Like that's incredible that the tick evolved this.
Oh yes. Well, we've been working on one of the compounds, we call it BSEP1. This was a collaboration with Ben Mans in South Africa. And it appeared that this compound has multiple functions. So it inhibits the complementary pathway, but it also has an influence on your blood coagulation. So, and that's all in one protein. So that's and that's also what Amine now discovered with one of his proteins, which he took to our lab. He also sees that one protein has multiple functions. So that's really intriguing.
Right.
Yeah, but one of my favorites is still evasion 3. That's because of the synthesis. It's easy to do, and it's really very stable. Yeah, it has an inhibitory cysteine knot, but yeah, we can cook it up to 100 degrees and for a couple of hours, and it's still there. So yeah.
Have you studied it? Because I know in one of the papers you'd kind of compared it to like linaclotide. Have you looked at the like gut stability of it? Even in just
the gut
like
stability? No, we didn't do that.
Okay.
That's a good one, and actually that's something I would like to proceed, because in the beginning we talked about the imaging studies. I did ex vivo studies, but I'm also trained as a radiochemist, so it would be great if I could radio label these compounds and inject it into a whole animal and see how stable it is indeed, and how fast it's cleared out of the body.
Right.
And whether it's cleared via the kidneys or via the liver, and whether we can really visualize inflammation in a whole body.
Right. Oh, wow. Do any of these proteins get taken up into any of the cells? Like, are they actually endocytosed, or is everything on extracellular activity?
Yeah, that's a good question. Maybe Amine can answer that one as well. What we saw with the evasions is that they really block by binding to the chemokine, and therewith they block binding of the chemokine to the receptor. So that's how they work. So that's mainly extracellularly.
Right.
Yeah.
Yeah.
Maybe
yeah. Sorry, go ahead, Ingrid.
So, Ingrid, you want to add something?
Yeah, no. I was asking whether you know from your experiments a little bit more whether tick proteins are going into the intracellularly into the intracellular environment or not.
Actually, the current projects we're working on and are these proteins that focus mainly on coagulation factors. So that's what we do in Ingrids Lab. So we have some proteins that are very specific to coagulation factors. Some of them are specific to factor 7 and tissue factor 7 complex. Some of them are specific to factor 10. Some of them are more up in the intrinsic pathways such as factor 11. So each protein is specific to one coagulation factor. And yeah, what's impressive here is that all of these proteins are together in the saliva, and then we see a complete blockage of the coagulation. And yeah, so that's what we currently are working on. Maybe I have no idea about the intracellular environment.
No, yeah, I think this is still something unknown as far as I know.
Yeah. Well, that's fun. There's more to learn.
Absolutely. Absolutely.
So, is there anything that I haven't asked that you wanted to talk about all these different proteins or different projects that you're working on?
No, not really. But what I would like to stress a little bit more, if I do have the time right now, is that in Northwest Europe and also in the US, we are actually only familiar with the notorious Lyme disease that are transmitted by ticks. And yeah, there's also tick-borne encephalitis, so you can get encephalitis. And in Eastern Europe, South Europe, people are vaccinated against tick-borne encephalitis. I don't know whether that's the case in the US, but there are many more tick diseases or pathogens that are transmitted by ticks. So you can get hemorrhagic fever. That's caused by a virus, Crimean Congo hemorrhagic fever. And there are multiple others, and these diseases really have a fatality rate of 30%. So it's really a lot. And it's not only humans that are suffering from tick-borne pathogens, but also animals, and especially livestock. Cows can get babesia or anaplasmosis, and also other fatal diseases. And you can imagine that if you are producing milk or cows, and that's a major part of your income, that ticks are really an economical burden for you. So yeah, I think that's important. A lot of people don't know. At least when I started in the tick field, I really didn't know this all. So
that's a great point. And so I guess, that in addition to looking at vaccines for humans, would this also be something that you would look at for livestock and whatnot to look for? Because I think now they dip the cows like in a right. Yeah, they should call it dipping or something like that. But yeah,
that's true. I've seen it myself in Africa and in Zambia, and it's really horrible to see because these cows don't like to go through it. It stings enormously. And it's only efficacious for 2 weeks, and then they have to go through it again.
Right.
Yeah, and then the meat is contaminated, and the ticks become resistant against all these chemicals.
Okay.
So so a vaccine for livestock would be great. That could be developed.
Okay. I might add something here, maybe some bad news for meat lovers, which is something else that ticks are doing, is that making people allergic to red meat, and it's called the alpha-gal syndrome. So if the ticks inject some sugar called alpha-gal in you, and your body makes reaction against it, then you are allergic to red meat. And a lot of people suffer from this alpha-gal syndrome. So more bad news, but we keep working on the good side of ticks, this gold mine called saliva.
Right. That's I didn't realize it, because I'd had that come up in my previous work when I was in the medical field, and I didn't realize that it could become that you could get the alpha-gal allergy from ticks. But that makes sense.
It's a possibility, and it's very bad news for meat lovers. Yeah.
Right. Yeah, we so I was telling Ingrid where I live right now. I'm surrounded by cornfields. I mean, I'm in the country, so there's lots and lots of ticks here. And we also have possums, and I learned that possums are very good for tick control because they eat them. So I guess either get a peptide or protein vaccine, or go buy like 1,000 possums to fit around your livestock.
It's also a good strategy. Yeah.
Yeah, I guess, something I do like to ask people before I close is sort of like, thinking ahead, like what you're hopeful for, what you think might be coming out of this work. And yeah, I'll just open it up. Ingrid, if you want to go first.
Mm-hmm. Yeah, if I really focus again on my research field, that would be that we can discriminate unstable plaques from stable plaques by an imaging method. And then I would really love to have a tick protein or a tick-derived peptide to make this possible, so that we can really see, okay, here a peptide is binding to inflamed endothelium, which means that this plaque is active, is unstable, and is prone to rupture. So that would be really fantastic, and of course something that could prevent a stable plaque to become an unstable plaque. Mm-hmm. But yeah, that's really far-fetched.
Yeah. That's a good goal, though. Yeah.
Yeah.
All right. How about you, Amine? What do you sort of, as you look at this, what are you hopeful for?
Yeah, actually, we are hoping for a lot of things. So we are working on different projects, and my hope is, I'm not going to say we will discover the most efficient anti-tick vaccines, but we are working on it. We also have faith in the proteins we are working on right now. So we really believe we could achieve something very advanced with these proteins, hopefully help with the therapy or diagnosis of some diseases, maybe cardiovascular or something related to inflammation or immune system. So that's what we are working on and hoping for. So my boss call it the bioticknology. So it's not biotechnology, biotechnology. So we're trying to do something with that, do something good for humanity and to help ticks to show their good side.
Right.
Hopefully.
Well, I'm very excited for the future of biotechnology.
Yeah. Well, the vaccines I also would like to work on, especially also the livestock vaccines, because I love animals. So yeah. But as you said, Wendy, that I was only allowed to say or to come up with one ultimate goal or ultimate dream.
Yeah. I would never limit your dreams.
Okay. Yes.
Well, yeah. Thank you both so much again. I really appreciate your time. I think this is an extremely fascinating world. I appreciate you explaining some of it to me.
Yeah, thank you as well, Wendy. It was very nice to have this chat and discuss the research. Thank you very much.
Yeah.
Thank you so much.
Thanks for tuning in to this episode of Exploration Science. As always, we welcome your feedback and suggestions for topics that you'd like to see covered. You can leave those suggestions in the comments below or tag us on LinkedIn. You can also find this podcast available as a YouTube series by searching Exploration Science. Thanks again for tuning in.